The Prix Galien is a welcome initiative to stimulate creative research and promote excellence. Barack Obama. The Galien Awards ceremony is considered the equivalent of the Oscars night for the innovators in the labs and awards every year the best pharmaceutical product, the best biotechnology product, the best medical technology, best digital health, best incubator accelerator, best startup.
This is the right event, on the right issue, at the right time.
I thank His Excellency, the President, for bringing Prix Galien to Africa, and I look forward to the day when we will celebrate an African winner of the Prix Galien.
I'm particularly grateful to receive this award.
The awards are among the highest honors in science and commerce because they lead to improvement in the human condition.
The Prix Galien Awards recognize the world's brightest minds and most innovative companies. They are a true celebration of the hard work required to produce life-changing interventions.
That is what makes us optimistic about the future.
Congratulations to all of you.
Make a difference. Join the Galien Foundation.
So welcome to the Galien Foundation Week of Innovation webinars here in New York. The objective this week is to provide additional content and background for our largest annual event, the Prix Galien USA Awards, to be held this Thursday here in New York. It's especially timely as the Galien Foundation marks its 50th Golden Jubilee year, representing 50 years of the best in new medicines research. And we've developed this new webinar series format to give our nominees for Best Pharmaceutical, Best Biotech, Best MedTech, Best Digital Solution, and Startup Accelerator Incubator categories far greater visibility in describing their journey of medical discovery. And this year's Galien USA Awards has a record 147 nominees for our 6 categories. And these all come from 126 different companies covering 15 diverse therapeutic areas ranging from cancer, cardiology and neurosciences to infectious and very rare genetic diseases and of course vaccines. And we'll be looking closely at the products in all of these critical areas throughout the week. Now, in the best pharmaceutical category, which is our focus today, we'll be holding 5 webinars due to the large number, 32 in total, of the products representing this important field. Now, this particular webinar in the hour that we have allotted is devoted to 2 nominees in the hematologic cancers and gene-based tumor agnostic therapies. And what we cover today relates to the central theme, which is, will the success of novel gene-based therapies in advancing survival timelines in blood and bone marrow cancers be repeated in the solid tumor setting? For example, with therapies that address each tumor's individual genetic profile rather than association simply with a particular organ in the body. Please note that the webinar includes some time for audience questions. So if you have questions, please state your questions in the link which is provided on your screen. But let's move right into the discussion for this first webinar today. And of course, I'll start by introducing the 2 nominees representatives who are going to join me in this webcast. So first, it gives us great pleasure to introduce Dr. Reshma Rangwala, MD, PhD, who is the Chief Medical Officer of Karyopharm Therapeutics, Inc. And Dr. Rangwala will be introducing the nominated product Expovio. And Expovio is a first-in-class oral exportin 1 inhibitor. And secondly, we're very happy to introduce Dr. Pam Kaur, who is a PhD and an asset leader at Servier Pharmaceuticals. And Dr. Kaur will be representing the nominated product Asparlas, and Asparlas is indicated as a component of a multi-agent chemotherapeutic regime for the treatment of acute lymphoblastic leukemia. So welcome to you both. And it's important to mention to everyone who's joined the webinar that there is an accessible link to a 100-word statement submitted by the companies representing each of these nominated products. So if you want to learn more about these innovative products, please follow the description link. But let's jump straight into the discussion, Dr. Rangvala and Dr. Kaur, about your products. And one of the questions that we have, which will help you to introduce the sort of innovative path that you've taken in discovering, developing, and bringing your products to patients, is why did your company choose to focus on the disease state and indication associated with the product? And in what ways did the product represent a therapeutic breakthrough, and how significant was the level of unmet need? And perhaps, Dr. Rangvala, you can start by introducing your thoughts on Xpovio.
Well, thank you so much. It's truly an honor, and on behalf of Karyopharm, we are very excited to be nominated and here to present Xpovio, a selective inhibitor of nuclear export, to the entire audience today. So again, I really appreciate the opportunity to discuss our compound with all of you today. It's a great question. Why did we go into multiple myeloma and diffuse large B cell? I want to start with, just our science and our mechanism. So Xpovio, also known as selinexor, is specifically a selective inhibitor of nuclear export. And as you mentioned, Paul, it targets exportin, also known as XPO1. Both the mechanism of nuclear export inhibition as well as its target modulates multiple activities that are really foundational to the growth and development of tumor cells, whether it's a hematologic malignancy or a solid malignancy. And I know we'll get into some of those details later. But, at its core, it modulates multiple key functions, including tumor suppression and that retention within the nucleus. It modulates oncogenes, it modulates DNA repair, glucocorticoid synthesis. So these multi-functional components allows us to leverage SINE inhibition across multiple different tumors. Based upon a decades plus worth of research, including both preclinical and clinical, multiple myeloma as well as diffuse large B cell, two indications that are currently approved in the US as well as 40 other countries, really suggested that we could harness selinexor to treat these very hard to treat patient populations. Despite, the multiple therapies that are available in multiple myeloma and diffuse large B cell, there remains high unmet need patient populations. And selinexor has really demonstrated benefit, for these patient populations, either alone or in combination. But this is really just the beginning for selinexor. And I will say there is another XPO1 inhibitor that we are also developing, eltanexor, that potentially allows us to broaden our availability of this drug, again, in other hematologic as well as solid malignancies. So the way I look at it is that we're just at the very beginning. And we really hope to see sort of able to transform the landscape and embed selinexor across multiple different tumor types.
Okay, thank you so much, Dr. Rangvala, for introducing your nominated product, and perhaps the same question to Dr. Kaur.
Thank you, Paul. Thank you as well for having the opportunity to share about Servier and its brand Asparlis. So to give you a little bit of background for the questions that you posed towards me, Servier already had an asparaginase product on the market. It was called Oncaspar. And it's been approved by the FDA for acute lymphoblastic leukemia for more than 15 years. Now, since then, we've been committed to developing the best asparaginase to remain the cornerstone of that multi-chemotherapy regimen for the treatment for it. Now, Asparlis, which is a type of asparaginase, it works by breaking down asparagine that's circulating in the blood, so the leukemic cells can't get what it needs, thereby killing the leukemic cells due to a depletion of the essential plasma L-asparagine. The second question that you had was, in what ways did we represent a therapeutic breakthrough? So the Servier Group is a global pharmaceutical company, and it's governed by a nonprofit foundation. And that enables us to keep patients first. We are able to reinvest in research, and it's allowed us to move the needle for those patients with an unmet medical need. And this commitment is why we brought Asparlis, and that's a more stable formulation of the asparaginase to market to ensure that every patient has the access to a potentially life-saving molecule.
Okay. Thank you so much. And in addressing, I guess we have the challenges that we all face in bringing innovative drugs towards patients. We have another question with respect to your two innovative products that you've described, and that is, what if any, and I'm sure there were many, what challenges did your company confront in transitioning from early proof of concept to human trials and then ultimately to market authorization? Like, what challenges did you face there? And when you think about those challenges, were there any lessons from your experience that you might want to add to really allow a better public understanding or awareness of what really goes into medicine, the medicine innovation process? Any thoughts on that? And maybe we could start with Dr. Kaur. How was the innovation process for asparagus?
Yeah. So, as I mentioned before, Servier had the asparaginase product in the market in Oncaspar, and it was approved for over 15 years. However, due to its complex manufacturing process and the significant supply chain challenges, we recognize the importance of bringing a more stable asparaginase to market for the patients who rely on this potentially life-changing molecule as their standard of care regimen. And I can attest that the supply challenges I deal with on a daily basis. So we've been able to provide this molecule. We feel that it's the best one for patients because it has a longer shelf life. It facilitates the management of product inventory and accessibility. It offers also a longer interval between doses compared to other available pegylated asparaginase products.
Mm-hmm. And in terms of the lessons simple lessons which we could communicate to the public about the challenges around the medicine innovation process, how would you boil that down into a simple statement, perhaps?
Yeah, that science is ever evolving, and this exemplifies the importance of being agile in order to find a solution for the best for patients.
Fantastic. Thanks Thanks so much for that, for those inspiring words. And to Dr. Rangwala, same question. Challenges along the road from discovery to market authorization, and lessons learned for XPOVIO.
Yeah, I appreciate the question. And Dr. Kaur, something that you mentioned at the end really resonates, right? I mean, sort of our approach to clinical development is ever evolving. And the reason I say this is because, sort of in the early days of selinexor development, right, sort of from a clinical development standpoint, we took a very traditional approach, right, of a 3+3 dose escalation. You identify the maximum tolerated dose. And ultimately, when we identified those doses, those doses ultimately became the doses that we took forward in the larger registrational, studies. With that said, right, we are very much informed by the patient experience, the physician's experience in dealing with their patients, real world evidence. And it's become acutely aware to us that toxicity has been limiting for some of these patients who are treated with selinexor. It's also enabled us an opportunity to potentially look beyond that and look to see how do we optimize that benefit risk. Can we potentially lower the dose, enhance the efficacy, but again, improve that safety and tolerability. Again, overall improving that patient experience, which is what ultimately, we aim to do. And so, again, transitioning from that traditional paradigm of 3+3 to now in a relatively short period of time, identifying doses that are potentially half, maybe even a quarter of the dose of where we started really allows us to not only improve the patient experience for those patients who are treated in multiple myeloma and diffuse large B-cell, but also to give us a foundation in which we can continue to develop selinexor in multiple other tumor types, including endometrial cancer, myelofibrosis, and other indications in myeloma. So it really has been a process. And, this isn't something that just our healthcare professionals have been asking, certainly our patients need. But we know this is an ask from our regulatory agencies too, that we transition in our approach to clinical development and really start focusing on this minimally effective dose instead of, again, the traditional maximum tolerated dose. So this is something that at Karyopharm, we are very interested in. But ultimately, at the heart of why we are so interested in this endeavor is because at the heart of it, it ultimately serves the patient.
Thanks so much for your words on that important topic as well, Dr. Rangwala, and maybe just to follow on from where you ended there, which is bringing innovation to the patients. I mean, another question would be like, what is the measurable sort of impact that you can cite on clinical outcomes for patients with your particular product? You've highlighted some already, but are there any specific things you would like to mention?
In our development programs, we by and large look at sort of the traditional endpoints, whether they're the surrogate endpoints of overall response rate, quality of life. Ultimately, though, I will say those ultimately serve to better identifying how we improve that survival in the patients, right? That is the key, is how do we improve the overall survival, but also, maintain that very important quality of life and that safety and tolerability. Ultimately, what we want to do is to maximize that benefit risk, turn these diseases from acute diseases, right, to a chronic disease that the patients absolutely can live with. And we don't want to burden the patient with undue toxicity that can sometimes be quite limiting. So that is very much, sort of how we approach our development process, again, in our currently improved indications, but in future indications as well.
Okay, thank you. And the clinician's response to the product so far, can you say anything about that?
Yeah, it is very much improving. I think, our focus on improving the patient experience, right? Improving that tolerability, profile resonates with the patients, with the physicians as well, right? I mean, ultimately what physicians want to see is benefit with their patients, that they're able to tolerate the drug, they're able to stay on drug. Both of those two parameters ultimately are going to drive efficacy, if not overall survival. So, you know, it is improving as they gain greater experience with the drug. They, you know, gain experience with the necessary prophylaxis, but they also gain experience with, you know, how to optimize the dose. It ultimately is, you know, giving them a confidence that they can treat their patients appropriately.
Thank you so much, Dr. Rangwala, for sharing that. And maybe a similar same question to Dr. Kaur in terms of can you cite, you know, the measurable impact that you've seen on clinical outcomes for the patients with this product, and any feedback from the clinicians with respect to your product this far?
Yeah, I mean, as I mentioned, asparaginases have been a cornerstone treatment for a multi-chemotherapy regimen, and we're proud to offer that this is a more stable form as a standard of care treatment option in acute lymphoblastic leukemia. So the NCCN, or the clinical practice guidelines in oncology, as a reflection of the clinical response to the treatment, it continues to recommend regimens that include a pegylated asparaginase as part of the induction regimen for the treatment of T-cell ALL in both pediatric and young adult patients. So as part of a multi-component treatment regimen, Asparlis helped patients achieve remission and long-term survival. So I can cite a few outcomes for you. We've observed 98% complete remission rates in patients with B-cell ALL treated with Asparlis as part of a multi-component chemotherapy regimen in a Dana-Farber Cancer Institute study. And an 87.6% had a 4-year overall survival rate when treated with Asparlis as part of a multi-component chemotherapy regimen in a Children's Oncology Group study. Now, we've had consistent and favorable disease-free survival outcomes as well in another COG-run study. There, 3/4 of patients treated with Asparlis were able to achieve minimal residual disease negativity by the end of their induction phase of treatment. So that gives you a little bit of an understanding as to what Asparlis's clinical response has been.
Thank you for sharing that, Dr. Kaur. Those are fantastic data. I think we have asked you and Dr. Rangwala a few questions there about your product and the experience in bringing your innovative products forward. Maybe now we can open up a little bit to a bit more of an open discussion, and certainly we'd like to follow this theme about how we can, you know, transition a lot of the successes in hematologic cancers into solid cancers, and maybe I could pick up on a point that you had mentioned, Dr. Rangwala, when you were describing Xpovio, which is indicated currently for you mentioned multiple myeloma, but that there was a lot of potential to bring that innovative product to other cancers, including solid tumors, and maybe and this is again the central theme of what we're trying to cover. So maybe we could have just an open discussion, thoughts. It would be great to hear your thoughts, Dr. Rangwala, on how on some of the challenges and how you see the field developing in terms of bringing agents into solid tumors from hematologic, some of the challenges, as well as obviously the potential in that large high unmet medical need group. Any thoughts?
Yeah, absolutely. So I go back, to the science and sort of the mechanism of selinexor and again, eltanexor, both of which are selective inhibitors of nuclear export. These are foundational again, right? Sort of these are tumor agnostic mechanisms again that are critical for the growth and development of cancer cells. We've shown the benefit in multiple myeloma and diffuse large B cell, but because we know that this is so foundational, there has been a lot of interrogation, both clinically as well as preclinically in solid tumors as well. And I'll give you a great example. So we've identified and, done a lot of work evaluating selinexor in endometrial cancer, for example. Endometrial cancer, just to step back here, right? By and large, patients who are recurrent or newly metastatic, they're treated with a very traditional chemotherapy regimen, so carbotaxol or a platinum taxane kind of regimen. They're treated with a fixed number of cycles, and once they complete those fixed number of cycles, NCCN guidelines recommends a watch and wait approach. Unfortunately, we know that many of these women, unfortunately, will relapse. And when they do relapse, their prognosis is poor. So what we have been studying is looking at selinexor specifically as a maintenance therapy. So again, once these patients complete that carbotaxol, then give them the opportunity to be treated with selinexor, again, as a maintenance in order to determine if we can improve and prolong their progression-free survival. We initially conducted the SIENDO trial, which was a randomized phase 3 study comparing selinexor to placebo as a maintenance therapy in these first-line or advanced endometrial patients. And what we observed was a very interesting subgroup analysis. Specifically, patients who are P53 wild type demonstrated the greatest benefit with selinexor. They actually had a 13.7-month improvement in their PFS as compared to only 3.7 months that was observed with placebo. But now contrast that to what we saw in the mutant population, the P53 mutant. If you look at that subgroup, there was no difference in the PFS between selinexor versus placebo. That data, and I will say, P53 wild type is identified in approximately 50% of endometrial patients. So this was a very robust subgroup, but those data also suggest that we've identified a very important biomarker in P53 wild type that can identify patients who best benefit with selinexor. I give you that as a background because it demonstrates that selinexor, yes, absolutely has an opportunity to benefit solid tumors like endometrial cancer, but we can also leverage a biomarker to better identify the patients who can benefit. So these are additional areas that we are actively investigating, not only additional tumor types in which selinexor benefits, but how do we leverage precision medicine and biomarkers to, again, find that right patient population who can benefit from this novel mechanism like selinexor.
Thank you. And does that, do you think you could use that biomarker P53 to consider more like a tumor agnostic approach and try and again, it was one of the central questions. Can we look at this in a tumor agnostic way?
And that is something that we are actively looking into, right? I mean, why, everybody asks why P53 wild type, and it's really not surprising. We know that P53 is very important in endometrial cancer, but if we go back to the mechanism of selinexor, right, P53 we all know is probably one of the most important tumor suppressors. SINE inhibition retains that tumor suppressor within the nucleus and therefore exerts its cell kill. So it very much latches on to the mechanism of our compound. To your question, can we leverage this across other tumors? Again, an active area of interest, and hopefully we'll have an opportunity to further study that in the near future, I should say.
Okay, thank you so much for sharing those important insights and exciting information. And Dr. Kaur, any thoughts on this from the perspective of Asparagus?
Yes. In fact, we've got some interesting preclinical data sets that we've been inching towards going forward with some solid tumors as well. And the same mechanism of action that we've seen that allows us to, killing leukemic cells that basically breaking down the asparagine that's circulating in the blood and depleting the L-asparagine allows for the kill for leukemia. It would also be the mechanism of action for killing in solid tumors. So those tumor types where they are dependent on L-asparagine would be targeted. So these are endeavors that we're seeking to go towards. Very exciting times for this molecule that's been on the market for many years. It's breathing a new life into the molecule.
And thanks for sharing that. Also, very, very interesting developments. And anything in terms of the development from hematology and hematological indications, solid tumors, the differences there. We heard about biomarkers for XPOVIO. Any other additional thoughts on going into the solid tumor indications that bring maybe additional challenges, Dr. Kaur, for Asparagus, or how would you approach that?
Of course, developing a biomarker strategy is everybody's mechanism to get to the goal, and that is a difficult path to go on. And so we're working diligently in order to get that worked out on our end as well.
Thanks for sharing. And so, Dr. Rangwala and Dr. Kaur, do you have any additional questions? The central theme of our discussion here today that you would like to share or bring up?
I will say, what we do from a clinical development and our ability to expand selinexor to the widest range of patients possible is to really look at it from multiple different approaches. So, we talked briefly about, identifying the right tumor, right? Whether it's a solid or a hematologic malignancy. Also, again, biomarkers and identifying the right biomarker and the right tumor type. But we are also very focused at Karyopharm on two other areas too, which include the rational combinations, right? So one of the areas that we can leverage is that we know that selinexor has both monotherapy activity in some indications, but also combination activity too. And I'll give you an example in multiple myeloma. Right now, we are approved in combination with dexamethasone as well as in combination with bortezomib, dexamethasone, but have also demonstrated very encouraging results when we combine selinexor with pomalidomide, dexamethasone, with daratumumab. So it really demonstrates the breadth and our ability to be able to exchange selinexor across multiple different backbones and therefore expand the patient population that can benefit from selinexor. The other area that I'll briefly comment on is that it's really important from our perspective to really understand sequencing of therapies, right? So we know, with this, fast emerging, mechanisms within the oncology landscape, to really identify sequence is going to be critical. So again, going back to some of our data, we've demonstrated in multiple myeloma that treating patients post anti-CD38 really has a potential to enhance efficacy. So understanding the association of selinexor post CD38, and then again, right, with the emerging, T cell and immune therapies in myeloma is again really going to be so key. Can selinexor maintain a normal immune environment so patients, if they wish, can then go on to get one of those immune therapies without compromising their ability to get harvested, et cetera. So, multiple different areas that we're focusing on, again, to best demonstrate that efficacy for our patients with cancer.
Thank you, Dr. Rangwala. Great insights, and I'm assuming that there's a lot of preclinical work that can be done to help understand the sequencing you mentioned and the right combinations for bringing the right combinations of XPOVIO to patients.
You are absolutely correct. And I will say we don't do this by ourselves by any means, right? I mean, we have so many different collaborators who are focused on the preclinical work, on the translational, work, on clinical work. And, that really is, across all of those different platforms, really how we continue to develop selinexor quickly, right, across all of these different indications. But yes, each one of these, and I will add to that, even real world experience, right, is really so informative and helps us identify, sort of the paths that we should pursue.
Yeah, great insights. And Dr. Kaur, for Asparlas, similar thoughts on combinations and sequencing, as we've heard from Dr. Rangwala, or preclinical work, translational work. What's your experience or thoughts or plans?
Yes, we've got many things ongoing with Asparlas as well. We're not only concentrating on the solid tumors, but also looking at other hematological malignancies that we could look at where this simple mechanism of action may be relevant. So it's quite interesting from that perspective as well. But much to your point surrounding biomarkers and targeted therapies, once you have something like that in hand, it's a lot easier to go towards making sure that we're being able to demonstrate efficacy. So, that's where our crux of push is as well on our end, to go towards the solid tumors with something in hand.
Mm-hmm. So combinations.
Combinations. Once we've demonstrated something in monotherapy, which we've demonstrated with our preclinical work, that is something that we're thinking that would be on the horizon as well. First, I think that the current data sets point more to even a monotherapy based regimen for solid tumors, which is very exciting.
Absolutely. Very exciting. Do you have any additional thoughts to the discussion, or any things that you would want to bring up that we've not already covered in our discussion so far, Dr. Kaur?
No, I think that it's, there's many people that are contributing to this, much like Dr. Rangwala had suggested. There's, we're going through and having that collaborative spirit and trying to combine with biotechs as well as academicians. It's not going to get done only by ourselves. So these are things that Servier is also committed towards in order to get that thought process going.
Thank you so much. It's so great to hear both of you say, and I can only echo what you say. Science and innovation is a team sport, and in bringing these innovations to patients, we have to work with innovation across the board. So that's great to hear. Dr. Rangwala, any additional thoughts from your side in terms of our panel discussion here on the key topic?
No, other than, I just want to say, I think, for us at Karyopharm, we're just at the beginning, right? I mean, there's so much opportunity, right? And again, we are laser focused on trying to maximize the opportunity for patients with our platform, right? With selinexor and then again, eltanexor, and continue to expand the number of tumor types in which we can do so. Yeah, it's great to be part of the company.
Thanks. Well, I would maybe have another question. So thinking to the future. So we're scientists and clinicians. Where do you see the next wave of innovations trending in your individual areas? And for example, what would the Galien Awards Committee be perhaps reviewing in the next five to 10 years? Like, where do you see the next level of innovation? Dr. Wangwala, maybe.
You know, I go back to a statement that I said earlier that, you know, patients with cancer have chronic diseases, right? Just like diabetes or cardiovascular disease. And if we want to turn this into chronic diseases for all patients with cancer, it's really trying to identify, you know, how do we continue to best sequence these therapies, right? So another area that we need to, you know, further invest, not only in Karyopharm, but across, you know, sort of all of our spaces, is to really identify areas of, you know, or mechanisms of progression. So unfortunately, right, we are not curing patients yet. We're curing a few patients, but I think, you know, we all want to expand that pool of patients who are becoming cured. And so really identifying what are the mechanisms of progression, how do we best identify those mechanisms, and then really indicate, okay, based upon those new mechanisms, mutations, et cetera, what is the next therapy that we can immediately put that patient on? You know, I look at it as that we are treating a patient, you know, they're going through a cancer journey, and we really want to be, you know, with them throughout their entire journey. So if we start them with selinexor, for example, you know, can we continue to follow them over the course of the multiple therapies that they may need, right, to keep their tumors at bay? So, you know, to build upon that, it's not only finding the right tumors, the right sequences, the right combinations, the right biomarkers, but also identifying when selinexor may not be working optimally, and how do we switch it out so that we can continue to improve and moderate their cancer.
Okay. Thank you. And Dr. Kaur, any thoughts on the future?
It's an extremely exciting time for the industry and for Servier. I think we're truly in the golden age of innovation, especially within oncology. With the emergence of targeted therapies, we're approaching new frontiers in cancer care that we haven't been able to do before. So this is, for Servier, what's guiding our innovation and reflected in our commitment to treat these hard to treat cancers, and also fulfill the patient's unmet medical need. We've had our robust oncology pipeline currently has 22 oncology projects in clinical development and 17 projects working on immuno-oncology, which are, you know, to activate the immune system against cancer cells, and targeted therapies to specifically target one of the biological mechanisms of cancer cells. So we have many, with that being said, we've got many avenues that we can pursue. And, you know, as I mentioned before, for Sparlis, it's exciting for solid tumors as well as other heme malignancies. Now, to your thoughts surrounding the crystal, you know, the 5 years, I'll say this: I wish I had a crystal ball. So hopefully we have cured cancer by then in 5 years, and we can say it's done. But I think that in order to get to that goal, clinicians, academicians, pharma, we know that this is too complex for, you know, any single organization to tackle on its own. So it's going to take all of us together to put this behind us, and in order to move towards that common goal. So Servier, as I mentioned before, has encouraged other pharma to drive that kind of wave of innovation by having collaborations with biotech companies and academics and startups. Now, through these collaborative partnerships, I see more potential treatment options coming forward where none existed before. And that's, going to provide cancer diagnosis options for patients. And that I think is key. So as a reflection of that commitment, Servier, we've formed about 40 research partnerships and collaborations to reach that goal. But hopefully, like I mentioned before, it's behind us and we don't even have to go just to the 5 years.
Thank you, Dr. Corey. You brought up a great point there of, the next wave of innovation may be very, very important combinations. Combinations of cancer cell targeted therapies with immune therapies is, obviously a very exciting area being progressed preclinically and clinically. And maybe you can comment a little bit on perhaps some of the challenges of those particular combinations, certainly in clinical development, the sorts of studies we'd have to put together to bring that level of combination innovation to patients. Any thoughts on that, since you mentioned it's a very important point?
Yeah, I think that Dr. Rangwala had already touched upon some of these where we've got to think about innovative design, sequencing. These are things that we're all grappling with within the industry. So in order to kind of go forward with that, there's many technologies that are modeling and simulation are there in order to kind of help us inch towards that. It's an exciting time through the innovation that we'll be able to leverage some of that towards the clinical aspects as well.
Mm-hmm.
Dr. Rangwala, do you feel the same?
I completely agree. I'm thinking about it too. Even just, the immunotherapy landscape really has, evolved, right? Just the number of different agents, whether it's from CAR Ts to these bispecifics, the anti-CD3s, etc., right? Each one of those may impact the patient differently, right? So trying to find even rational combinations within the immunotherapy space is going to become a very important area of work, right? Really understanding, sort of how these immunotherapies may have long-term consequences on the immune milieu in the patient is going to be important, especially as we try to understand, right, again, that sequencing component, right? What are the next best therapies that the patient should be treated either pre or post those therapies? The other aspect that I think that we all need to be focused on is that, some of these immune therapies unfortunately have a lot of toxicity, right? Including the CRSs, neurologic phenomenon, and may not be available to all patients as a result. So even trying to identify the patients who are going to best respond to those immunotherapies and can receive those immunotherapies, to say nothing about the rational combinations then that we could pursue within that subset of patients. So a lot of more questions, unfortunately, than answers, but it's a great place to be in because it ultimately means that we've got more therapies ultimately for the patients.
Most definitely agree.
No, I think that was a great discussion, and to the question where we see the next wave of innovations, I think it's clear from your answers that that's certainly going to be in the right combinations, and that's a key theme, obviously in the oncology space. Now, I'm looking to see if we have any questions from our audience. I don't see any open questions. So if you've got any questions that haven't already been addressed for the panelists, if you've got any questions for us, please pop them now in the chat there, and we will be able to see them. But I think we've already covered some of the key points about where do we see the status of your innovative nominated products and what's the future. Translating them into solid tumors is clearly the next step, and you've highlighted a lot of the advantages and plans and challenges you will face in doing that. And I think that's addressed one of the key topics that we had for this opening webinar of the series. And I don't see any open questions. Any final thoughts, though, from the panelists, Dr. Rangwala, Dr. Kaur? Any final thoughts from your side? Anything that you'd like to bring forward about your nominated products, or anything that we've not covered that you think is important?
I just want to emphasize that we're truly honored, at Karyopharm, again, to be nominated for the Prix Galien Award. And, it just gives us it's a morale boost, right? As we continue to develop this important therapy. So again, I just want to, say thank you on behalf of all of us at Karyopharm.
I echo your similar thoughts, Dr. Rangwala. It's been an honor to be nominated for this award, and truly helps us, gives us as well as some morale boost to help drive us, and open our laptops every morning to keep going. There's much to be done, as we've mentioned, between the three of us. Some days it's overwhelming, and some days we keep striving. This really does provide a lot of boost to us, and thank you for the nomination on behalf of Servier as well.
Thank you so much to you both. I think we've had a great discussion. And I would wrap up our panel discussion now, and thank you both so much for the excellent discussion, addressing some very key hot topics for introducing your nominated innovative products. And thank you so much, Dr. Rangwala and Dr. Kaur, for your input to this important theme of this particular webinar. And I would end, unless there's any late breaking questions, which I don't see. For thanking everyone for joining us, and also very importantly mention that this webcast is viewable on the Galien Foundation's homepage. And we would also like to highlight Thursday's Jubilee Forum, again celebrating 50 years of innovation in new medicines. And this will feature a group of CEO keynote speakers, as well as the presentation of the 2022 Roy Vagelos Pro Bono Humanum Award winner, and that will be at 6:30 p.m. New York time. And this, of course, will be followed by the awards gala. And both of those events, of course, will be held at the American Museum of Natural History. So from Dr. Rangwala, Dr. Kaur, and myself, we thank you all for joining this webinar, and we wish you a nice day. Thank you.